This recording discusses genetic inheritance patterns that are beyond simple complete dominance. Just as a reminder, a dominant allele does not actually interact with or repress or anything like that the recessive allele. They're completely separate. It's just that they code for different protein products or different regulatory products because of their different DNA sequences. And that just means that depending on how much of the product you get expressed, like if you have two alleles that express it, or two alleles that don't, or one allele that does and the other one that doesn't, that's going to give you your phenotype. It's also a misconception that dominant means the allele is more common in the population. That's not necessarily true. For example, polydactyly, having extra fingers or toes, is a dominant allele, codes for that. So that means you only need one copy of that allele to have extra fingers or toes. But obviously, it's a rare trait to have. So the dominant allele is rare in the population in that case. Dominant's also not necessarily better. It depends on the condition. So it just means that you just need one copy inherited in order to see the phenotype. That's all that dominant refers to. So beyond just looking at a single gene and complete dominance, we have other inheritance patterns. For example, certain alleles are not completely dominant or recessive. We call them incomplete dominant or codominant, and so we'll talk about those. Many genes have more than two alleles. In fact, almost all human genes have more than two alleles. And that means that different combinations can lead to different phenotypes. And there are also genes that produce many different phenotypes just from one gene. And so that's going to also have different inheritance patterns and consequences. One type of inheritance pattern that's not complete dominance is called incomplete dominance, which is basically what it sounds like. It means that neither allele covers up the other allele. But in this case, when the heterozygote, both alleles are present, it's actually a new phenotype that doesn't look like either parent. And usually it's some sort of blended intermediate phenotype. So a classic example of this is snapdragons. So if you take a red snapdragon and a white snapdragon and you cross them, you actually get all pink snapdragons. Now if Mendel had used snapdragons and he stopped there, he might have actually thought that the inheritance was blending, like that idea that people had, that the red and the white kind of went away. But when you take two pinks from that generation and cross them to each other, you actually get red and white back. And in fact, you get one red flower for every two pinks for every one white flower. So this should look like a pretty familiar phenotypic ratio, the 1 to 2 to 1, but it really looks like what we've seen genotypically, where you have three different categories. So far, our heterozygous, our monohybrid crosses have been 3 to 1 phenotypically. This is a 1 to 2 to 1, and let's look at why. So when we write alleles for incomplete dominance, it's not technically correct to write big letter, little letter, because big letter means that it's dominant and incomplete dominance is not dominant. So one way to do that instead is to write something like A1 as allele number 1, like A1 codes for red and A2 codes for white. So A1 allele, A2 allele. You can also write things like A allele and A prime. C, R, CW is for red and white, but it could be any combination thereof. But it's just indicating that you have the same gene, the C gene, and there's two different versions of it, neither of which is dominant. The A gene, two different versions of it. And here's another way to write that. So what would we see in this cross here? So we'd see that red is going to be homozygous for our A1. White's homozygous for our A2. Pink is our heterozygote. And then when we cross it again, it's just like the same old monohybrid cross, but since it gets us the same ratio, 1 A1 A1 to 2 A1 A2s to 1 A2 A2. But in this case, unlike the complete dominance where these two guys look the same, now the heterozygote has its own phenotype. And that's really the key feature of incomplete dominance, to recognize that. A human situation where we see incomplete dominance in health conditions, a genetic disorder actually, is looking at these receptors for LDL. So low density lipoprotein is the LDL. The receptors are supposed to take the cholesterol protein complexes out of the body. They bring it to the liver in order to get removed. And so most people are big H, we'll say. So this is allele number 1. I actually don't like the H's like that, but we'll say they're A1. So they make a lot of the LDL receptor because both genes code for it and both genes make protein. So on their cells, they have full complement of the LDL receptor. Then there are some individuals who are A2. Now we'll say that A1 codes for the receptor. A2 is broken. It codes for no receptor, which means that their cells have no receptors on them, which means they can't remove the cholesterol, and they have very high cholesterol levels that are very dangerous. The heterozygote, an A1, has half of its DNA has a functional receptor-forming gene, and half its DNA does not. So in this case, it actually matters phenotypically. So on the cells, they have half the amount of the receptor protein, which means they get an intermediate phenotype in here. So these hets have an intermediately high cholesterol levels, which can actually be dangerous as well. So the genetic disorder that works like this is called familial hypercholesterolemia, and it's an example of incomplete dominance. So another type of inheritance pattern would be when you look at multiple alleles. This is, again, for most genes in the population, especially in humans, and one example we use a lot is the ABO blood group. So this is the blood types when you usually think of blood type. Although this is only one type of blood type, there are other ones too. This is the most commonly referred to one in addition to the positive negative because it's important for blood transfusions. So here is the what's called the gene, the I gene, and it encodes an enzyme, and there's different versions of it. There's one that attaches an A carbohydrate, there's one that attaches a B carbohydrate, and there's one that attaches no carbohydrates, and that's all to red blood cells. They are also what we call codominant, where that means that if you have both, so you're heterozygote, you're going to see both phenotypes. So instead of incomplete dominance where you see an intermediate, like a between, new phenotype, this one you're going to actually see in this case, the A phenotype, and you'll see the B phenotype at the same time, and that's what codominant is. So here's how you write the alleles for this multiple allele situation. We said it's the I gene, so the I superscript A, we can call it IA, allele codes for the enzyme that attaches this A carbohydrate. The I superscript B, the IB allele, codes for the enzyme that attaches the B carbohydrate. And the little I allele codes for a broken enzyme, so no carbohydrate gets attached. And you can see these can come together in multiple ways. So we can have homozygous versions of them. We can have heterozygous versions of them. I mentioned that there was a codominant situation here, so this is our codominance. You can see when you have a heterozygote with an IA and an IB allele, you see both the IA phenotype, the triangles on the white blood cells. You see both the AI phenotype, the triangles in the red blood cells, and the IB phenotype, the circles on the red blood cells. So that is a codominant scenario. You'll notice that there are 2 ways to get A blood. There's 2 ways to get B blood. Now, in this case, it doesn't matter the amount of carbohydrate you get on there. Phenotypically, these both look A. They both behave the same way. So it doesn't matter whether you have 2 copies or just 1 copy and the recessive. And you'll notice that the only way you can get no carbohydrates is to have 2 broken copies, 2 little i's. And phenotypically, these are the different blood groups, A, B, AB, and O. So A means that you just have the A carbohydrate, B, you just have the B carbohydrate, AB is the codominance where you have both, and O is completely recessive where you have neither. So let's work a problem with blood types and see how that turns out. And I'm actually going to use my family because we're a nice mixture of blood types. So I am type O blood. My brother is type AB blood. My sister is type B blood. My mom is type A, and my dad is type B. So let's see if this family actually works. So certain ones are easiest. So there's only one way to get O blood and only one way to get AB blood. So we know these are all phenotypes, but we know my genotype for sure. The only way to get O blood is to have little I. My brother, we also know for sure. The only way to get AB blood is to have IA, IB. Then for my sister, mom, and dad, there are two ways to get each of those. So my sister has to have at least an IB, and then she could have either another IB or a little I. My mom must have an IA, and she could have another IA or a little I. And then my dad's like my sister. He has an IB and either another IB or a little I. So let's see. You might be able to see it, but let's figure out if we can determine my sister, mom, and dad's blood types. So you can draw a Punnett Square and put in what you know. So we'll put my mom down the side, my dad on the top, and I'll be in here. So we already can distribute this because we'll say this is the egg and this is the sperm. Okay, so the only box left for me is over here. So that must be me. And that means what happens to my parents. So both of my parents must have had a little I because that's how I got it. I have to get one from each of them. So my dad must have a little I and my mom must have a little I. So we can fill that in. So we've already made a box that's my brother, the AB box. And then if we continue to fill that in, now typically we actually wouldn't write it little I, IB. We'd write this first and the little I second. But in any case, that one box is actually my sister. And my parents could also had a child with A blood type, with the IA little I. Now, if they had had a fourth child, would it for sure have A blood type? No. It's a 1 in 4 chance every time. So all my siblings and I, all three of us, could have had O blood like me. It's just a 1 in 4 probability each birth. You can also work kind of paternity problems where you have a given mother and a given baby and a potential father to tell whether it's likely the father or not. Now, of course, there are always weird situations that happen with blood, with any genetics really. So just because something's not working out doesn't necessarily mean that the father is not the father, but it just means that it probably needs to be looked into further because it's either something really weird or the father is not, or the paternity doesn't match up. So let's say we have the mother has A blood and the baby has AB blood and the potential father has O blood. And the question is, can these parents have this baby? So sometimes people can see things right away, but we'll write out what we know. So we know that the kid is, we know that the father is little I. That's how you make O blood. And that the kid is IAIB. That's how you get AB blood. And the mother has an IA and then either another IA or a little I. So there's our kid. We're trying to make this from the mom and the dad. So here's our mom. So that works out fine. Mom donated the big A, mom donated the IA allele. What did dad need to donate? We needed dad to donate the IB. And if we look at dad, potential dad does not have an IB. And so that means that this is unlikely to be the father of this child. Physically, this is one of the reasons that ABO blood type matters and why they ask you for your blood type for blood donations and things like that. It's because if you don't get the right blood type, then you can get a really bad reaction to it. Your body starts attacking the blood as a foreign invader and you die. And so why does that happen? That happens because the different carbohydrates on your red blood cells, if your body has not grown up seeing them, it will trigger an allergic or if your body has not grown up seeing them, it will trigger a inflammatory reaction, as in, this is a foreign invader we need to kill. And so if you have A blood, either genotype, that means that you have antibodies against B, which means you've never seen B before. So if you get anything with B in it, that's when you're going to have a reaction. But you can get A blood, so A can obviously get A blood. A can also get O blood because O blood has no carbohydrates. It's not going to react with anything. In fact, you'll notice that O blood can be donated to all the different blood types. B can get O, AB can get O, and O can get O. And that's because O doesn't have any carbohydrates on the red blood cells or it doesn't have any AB carbohydrates on the red blood cells to activate the immune system. You'll notice that AB blood has seen everything. It has no antibodies, so it can get any blood type. It doesn't matter if it's seen A, it's seen B, it's seen both of them together, and so there's no reaction. And in fact, this is actually why AB blood type is preferred for plasma, because the plasma is not the red blood cells, it's the liquid of the blood. And so the person who has AB blood actually has no antibodies in their liquid. So that's quite useful because that way if you give someone else the liquid, it's not going to have any kind of reactivity. On the other hand, AB blood is not very good for donation for whole blood, for having the blood cells, because you'll notice AB causes a reaction with A because it's never seen B. AB causes a reaction with B blood because it's never seen A. AB causes a reaction with O blood because it's neither seen either. So actually, O blood is preferred for whole blood donations. AB blood is preferred for the platelet donations. So also, it is important if you don't know your blood type to try to find that out because if you're in an accident and you're able to communicate and you need a blood transfusion, it's very helpful if you can tell them what type of blood type you have. If they don't know, they'll just give you O, which is why it's really great to be a donor if you have O blood type. But they'll take anything, and so donations of blood are very important. You might have also heard when talking about your blood type of a positive negative. I'm O positive. Someone might be A negative. And what does that positive negative mean? That stands for the Rh factor. So a positive means that you code for the Rh factor, a negative means you don't. So this was what a red blood cell would look like in a person who has the allele that codes for the Rh factor. So whether it's homozygous or it's a heterozygote, the red blood cells have the factor on it. In people that are Rh negative, it means they have two recessive alleles, so they don't have the factor on the red blood cells. So why does this matter? If you don't have the factor on your red blood cells and you get blood that has it, your body's not going to recognize that blood and it's going to attack it, just like with the ABO problem. So if you're Rh negative, that's really important to know because you have to avoid getting Rh positive blood. It's also important in pregnancy. If a mother is Rh negative and her developing fetus is Rh positive, the mother's body, their blood never actually mixes, but the mother's body can detect that there are red blood cells that are Rh positive in the woman's body. It doesn't recognize them and it attacks them and it can actually kill the fetus. It's called the hemolytic disease of the newborn. And so if you are a woman that is Rh negative, like you have A negative, O negative, B negative, or AB negative blood, then you just need to right away when you're pregnant get tested. And if your fetus is Rh positive, it's good news is that there's a shot that you can get a few times throughout the pregnancy that keeps your body from attacking the fetus, and so you can maintain the pregnancy and have that child. So now we've defined complete dominance, incomplete dominance, codominance, and we've said that this allele has this type of inheritance pattern. But it actually depends on what level you're looking at. For example, in Tay-Sachs disease, it's a fatal disease that kills in early childhood, and it's due to a dysfunctional enzyme causing accumulation of lipids in the brain. So the enzyme usually breaks down the lipids in the lysosomes. When that enzyme's non-functional, the lysosomes accumulate with the lipid in it, and the neurons die, and eventually the child will die. So I'm going to say T1 is going to stand for our functional enzyme allele. T2 will be our non-functional enzyme allele. So on an organismal level, somebody with T1 will look normal. They don't have Tay-Sachs. Someone with T1, T2 also looks normal. They don't have Tay-Sachs. Only someone with T2 has the Tay-Sachs and will die from it. So that is a recessive, where you have to have 2 copies of the allele to show the phenotype. On a biochemical level, though, when we look at T1, it's going to have 100% enzyme activity because both the T1s code for enzyme. If we look at T1, it actually has 50% of the enzyme activity because only one of those alleles is functional, coding for enzyme. But apparently in this case, having 50% enzyme is enough to keep you from getting Tay-Sachs. Only when you have T2, where you have no enzyme activity, is where you have the trouble, and that's where you get Tay-Sachs overall. Molecularly, and that is an incomplete dominant scenario where your intermediate phenotype is the heterozygote. Molecularly, the alleles are actually codominant. So T1, so T1s stand for an enzyme, let's say it looks like this. So all of your enzyme is looking like a circle. The heterozygote has half its enzyme looking like a circle and half its enzyme looking like not a circle. So this is the non-functional version, but it still produces something. T2 has all of its enzyme in the non-functional form. And so you see here the codominance is because you see both phenotypes. So this is one phenotype, this is the other, and then you see both of them here in the heterozygote. So usually when we're talking about things, we're talking about it at the organismal level. But sometimes if you start thinking about inheritance and you look at it at different levels, you could get confused as to, wait a second, but I thought that this was recessive or dominant or something like that. So just pay attention to what level we're talking about, and if it doesn't specify, it's almost surely going to be the organismal level. So pleiotropy is an important term to understand. Pleio means more, and the more is referring to more than one phenotype. So this is when you have a single allele that is coding for many different outcomes. It doesn't have to be a disease, but a lot of times we talk about diseases as pleiotropic because when we see simple Mendelian inheritance like sickle cell or cystic fibrosis or Tay-Sachs, usually it's not one single phenotype coming from that disease. So even though you have one problem, like in cystic fibrosis, you have little c, your only mutation is in that one gene, you have a slew of different phenotypes. Here the example of sickle cell. So sickle cell, you only have one mutation, but you have all these different phenotypes. So kidney failure, heart failure, brain damage, anemia, pain and fever, joint problems, all these kinds of things come from the fact of just one allele problem. So that's pleiotropic. Pleiotropic, one gene, many effects. Pleiotropy is kind of the opposite of polygenic inheritance. Polygenic means you have many genes, polygenic, but one effect. So most of the time, polygenic inheritance is talking about quantitative characteristics, which means that you build up the amount of alleles and you have this kind of continuum of inheritance phenotypes. So something like skin color, this is that example, height, tail length on an animal, stem length on a plant. It's not like discrete units. Someone's not just 5'2", 5'3", 5'4". I mean, that's what we say they are, but there's really all sorts of in-between phenotypes. And that's because there's many genes that are contributing to that one phenotype. So for this example here with the skin color, they're showing 3 different genes, the A gene, the B gene, and the C gene. And of course, there's 2 alleles of each. So you have 6 alleles total that are contributing to the skin pigmentation. And just each allele, simply, they have whether it's contributing lots of pigment, and they've colored that dark, or little pigment, and they've colored that light. There are different ways that the genes can contribute to that. Sometimes they produce enzymes that convert products into pigment or don't. And sometimes they do regulatory genes that turn off and on the amounts of the other genes that make the pigments. So there's all kinds of ways that these genes can result in pigment or not. In any case, we have 2 people here who are tri-allelic hybrids. They're heterozygous for all 3 of those genes. We cross them here, and you can see there's their medium skin tone, and they get a slew of all the different types of skin tones. Now, most probable would be having medium skin tone babies, but that doesn't mean they can't get a very fair child or a very dark child. Just depends on how the alleles get recombined and shuffled up. And so that's why you get this continuum, because many different alleles adding together in different ways gets you lots of different phenotypes. Here's an example of a polygenic inheritance type problem. The height of the spikeweed is a result of polygenic inheritance involving 3 genes. Each gene contributes an additional 5 centimeters to the height of the plant. The plant base height is 10 centimeters. The tallest plant can reach 40 centimeters, and that would be all dominant. So the plant starts at 10, and then each dominant allele gives you 5 more. So in that case, we can see 5, 10, 15, 20, 25, 30. So 30 extra centimeters onto the base of 10 gives you 40. So we say if a tall plant and a base height plant, the shortest plant at 10 centimeters, are crossed, what's the height of the F1s? So we know that when we get complete homozygous and dominant, complete homozygous recessive, that means that we get complete trihybrids here. And each of these, we have to start with a base of 10, and then any dominant allele we have adds 5 more inches or 5 more centimeters. So we have 1, 2, 3. So we add on 5 3 times, and that gives us 25 centimeters total. So a tall plant and a short plant are going to equal result in 25 centimeter plants. Now the question is, how many different phenotypes will be in the F2 generation? So you can do Punnett squares, but they get quite large. And so a trick is that however many alleles that you have, so here we have 6, if you subtract 1 from that, that will give you the number of phenotypes in the next generation. So if you do all the combinations, there'll be 5 different phenotypes that can come from a cross between 2 of these trihybrids. So we looked at 2 genes in those dihybrid, trihybrid crosses, but that was when the genes were doing their own thing. Now we're looking at genes that are working together to create a single phenotype. So we just looked at polygenic inheritance, and that's one example of that, where you have many genes in one phenotype. But there's also epistasis, is a situation where you have more than one gene contributing to a phenotype. Multifactorial characters are also when you're looking at multiple genes contributing to a phenotype, in addition to environmental interactions. The word epistasis means standing next to or standing on top of. And what this means is that you have at least 2 genes, and one of those genes dictates whether you see the other gene or not. So they don't physically interact. It's not like one shuts the other one off. But just the way that they work in the signaling pathway or the coding pathway means that if one has a certain genotype, then you will see the other one. If the first one has a certain genotype, you won't see the other one. So let's give an example, because it's sort of hard to explain in words. This is the classic example of the co-colors. So they have mice and dogs, and this is mostly true for most coat colors. So there are a black pigment allele we'll call big B, and a brown pigment allele we'll call little b. So this is a complete dominance scenario here. Then there's also another allele, so it's called C. C means color, and little c means no color. And what happens there is that in the big C, the machinery that deposits the pigment that's made into the actual hair shaft is working. In the little c, the pigment never gets from its origin into the hair shaft. And so when you have little c's, you'll never see your pigment color because it never gets to the hair. So it's there, it's just not seen. And that's what epistasis means. It means depending on what C you have, dictates whether you even see your B or not. So here's that color deposition example in both dogs as well as mice. And you can see they've done their 16 square Punnett Square, but it's a dihybrid cross. And remember, when we did a dihybrid, we expected a 9:3 to 3:1 ratio. That's what we saw with like the pea shape and the pea color, or plant height and flower color, whatever two you looked at before. You got 9 dominant dominant to 3 dominant recessive to 3 recessive dominant to 1 recessive recessive. But this looks different. It adds to 16 still, 9, 3, and 4 still add to 16, which means 2 genes are involved. But you'll notice that we've combined 2 of these together. And they can be combined in all sorts of ways. It just depends on the inheritance. But let's look at what happened here. And I'm actually going to do it looking at it not with the Punnett Square, but just with our probabilities, because that's the way you want to work it in the end anyway. All right, so we'll do it with the dogs. So these are labs. So we have black labs, we have brown labs, chocolate labs, and we have the yellow labs. Those are our 3 phenotypes. So let's think about how we can get those. All right, so black and brown are both colored, while yellow means no color. So in order to be black or brown, to show the color, that means that we must have the color deposited. So we have to have a big E as part of our genotype in order to even see that color to begin with. In order to be yellow means that we have no color deposited. So you know for sure yellow is little e little e. Now the tricky thing about yellow is you can't tell anything else about it, because when it doesn't deposit the color, you won't ever know whether it has big B's or little b's unless it has offspring. Then you could track backwards to see what the yellow has. So right now, all we could leave yellow as is anything with 2 little e's will look yellow. To be brown, brown's recessive, completely recessive to black, so that means we need little b to look brown. And to be black, we need a big B and something else. So if we do our probabilities, and you can do your Punnett squares, this is the time they're independently assorting, so you can just do your Punnett squares on their own. Should look familiar. Might be getting used to the ratios by this point, not having to write it out every time. Okay, so what's the probability of getting any big E? So if we look at here, 3 out of 4 of our boxes, so it's a 3 out of 4 probability of getting a big E. Probability of getting a big B, also 3 out of 4 in this case. So now we have 3 times 3 is 9, 4 times 4 is 16. There's the 9/16th chance of getting a black dog from 2 black dogs, actually. So both these dogs would be black. To get a brown dog, E again, big E is 3 out of 4. Whereas little b, there's just 1 box of that, so that's 1 out of 4. So that gives us 3/16th chance of getting a brown dog. To get a yellow dog, we need little e. There's 1/4 chance of getting a little e from this cross. And then we could anything else, and we'll still look yellow. So that's 100%, or 4 out of 4 boxes, or 1. So all boxes will work for that. So that gives us 1 out of 4, which translates to, or converts to 4/16. Now you want to make sure you add up to 16. 9 plus 3 plus 4 is 16. And so that means that, and that's probabilities. And if I asked you for ratios, then you said the ratio would be 9 black to 3 brown to 4 yellow. Here's another epistasis scenario, a similar coat color problem, but this one's a little bit different. This is in horses. There is an E gene, and we'll say big E deposits black pigment, and little e is red pigment. So anything with a big E is going to look black. When you have little e's, then you can see a second gene. So with little e's, there's actually there's a modifier gene of the red. So it's called the cream gene, CCR. So if you have CC, that is not modifying at all. So you just have full red color. You look chestnut. It's a type of horse coat. If you have a heterozygous genotype, meaning you have one C and one CCR, then you get some dilution of that red, and you get a gold coat, and that's called a palomino horse. If you have CR/CR, you get more dilution of that red, and you actually get a cream or white coat, and we call that cremello. So let's look at a dihybrid cross with two black horses. So actually, big E, little e gives you a black horse, no matter what else there is. And so that's how we know what those are. So we'll just do the crosses on their own. They're independently assorting, I should tell you that. And we basically, because these are dihybrids, they end up always being the same. Now, if we didn't have dihybrids and we had some other combination, you wouldn't always get this 1 to 2 to 1, but just pay attention. Okay, so let's see what we can get. So we could get black horses. We could get chestnut horses. It's completely red. We could get palomino. It's the gold. We could get cremello. That's the cream coat. So let's look at our genotypes we need for each of these. Okay, to be a black horse, big E and anything else will get you a black horse. What about a chestnut horse? Well, any of these guys are red based, so they have to have two little e's. To be chestnut, we said that's big C, or CC, full color. Palomino is partially diluted, CCR. And cremello is fully diluted, CCR. And so let's look at the probabilities of each of these. To get a big E, we can see here, that's 3 out of 4 probability. And then any of the other ones, which is 1, so we have a 3/4 chance of having a black offspring from these two black parents. A chestnut, so little e is a quarter. That's for all of these, actually. Probability of having big C, it's 1 out of 4 boxes there. So that's 1/16. Probability of having a heterozygous for the CCRs, that's going to be 2 out of 4 boxes, or 1/2, 2/16, 1/8. And then the probability of having CCR is 1/4, so that's 1/16. So we want to add this up, actually. So to convert 3/4 to 16ths is 12/16. So 12, 13, 14, 15, 16. Okay, good. So we expect 12 black horses to 1 chestnut, to 2 palomino, to 1 cremello. And that would be the proportions of the offspring that we're expecting. This would be the expected phenotypic ratio. So you can see this is a case where we have epistasis, and we also have, on top of that, this is an example of incomplete dominance there for the modifier gene. So there's lots of combinations. You just work it little bit by little bit, and if you just put it together in the end, you should be okay, as long as you don't panic. So here's another example of epistasis. Again, it's coat color, but this time it's in horses. So the way horse coat color works is that there's an E gene, and that dictates whether you have a black coat or a red shade of coat. So big E gives you black coat, and little e's give you a red coat. If you have a red coat, then there's a second gene that comes into play. And this one will modify the red color. So if you have CC, you get full red color. We call that kind of horse a chestnut coat. If you have a CCR, the cream allele, you're partially diluted red, which gives a gold coat, and that's called a palomino horse. And if you have two of the cream alleles, it's very diluted red, which actually looks cream or white, and we call that kind of horse a cremello. So let's look at the types of genotypes in order to get the phenotypes. So we can have a black horse, we can have a chestnut horse, we could have a palomino horse, and we could have a cremello horse. To get a black horse, we need a big E and anything else, both for the E's and the C's. To get a chestnut, palomino, or cremello, all of those are red-based, so red only comes out when you have two little e's. Then the modifier gene comes into play. So to be a chestnut, we have the CC, full red color. To be palomino, we have CCR, so one allele of the cream, so your gold coat. And then if we have two of those cream alleles, then we're cremello. So let's look at the probabilities of getting each of these genotypes from this cross here. So this is a dihybrid cross, as usual, of two black horses this time. But the black horses are heterozygous for both the E's and the C alleles. So they're independently assorting, which means that we can just look at our E's on their own, do a little monohybrid cross, and our C's on their own, and do a little monohybrid cross. Okay, so I've done the crosses. So to get a black horse, we need a big E and anything. So we have 3 out of 4 chance of getting a big E, and then 100% chance of getting anything else. So that gives us a 3 in 4 chance of having a black offspring. Chestnut needs 2 little e's. In fact, all these do. So little e is 1 out of 4, so it's a quarter chance for all of those to be a red-based horse. And then getting 2 C's is 1 out of 4. So that gives us a 1/16th chance of having a chestnut offspring. Palomino has 2 boxes for this heterozygous for the cream allele. So that's 1/2 or 1/8, 2/16. And then cremello only has 1 box as well, so 1 out of 4 gives us 1/16th chance of being cremello or having a cremello offspring. Just to double check, we'll convert this to 16ths. That's 12. And let's make sure this adds up to 16. 12 plus 1 plus 2 plus 1 equals 16. So if they asked us for probabilities, these would be our probabilities. What's the probability of getting a palomino offspring from these 2 black parents? That's a 1/8 or 2/16 chance. If they asked us for what's the phenotypic ratios expected from these parents, we'd say 12 black horses to 1 chestnut horse to 2 palomino horses to 1 cremello. That would be a phenotypic ratio of expected offspring. So that's an example of epistasis, because the E dictates whether you even see the red or not. And then on top of that, a dilution gene, and it's incomplete dominance. You'll notice that this is an intermediate phenotype, full, very diluted, and intermediate diluted. So you can have all kinds of combinations, but as long as they're independently assorting, you can just work them as their own separate Punnett squares and figure out with the blanks. It's going to be the easiest way to do that, and you just put in the probabilities for each of the different genotypes, and you'll find out the probabilities for the phenotypes. There's also many phenotypes that are impacted by the environment. So one of these classic examples are hydrangeas. So hydrangeas come in shades of color, so they come in pink and blue, or blue-purple. And what actually, it's the same, this could be the same plant, but if you put it into different soil acidity, it turns red or blue. And so that's, we call that the norm of reaction. So the same genotype can go all the way from a red phenotype to a blue phenotype, and so that range is the norm of reaction. Skin color is a good example of having a norm of reaction, because it can go all the way from very light all the way to dark, and even on the same person, actually, I should say, due to how much sun exposure. Your hair has a norm of reaction, depending on how much sun. People's hair lightens or doesn't. Your height has a norm of reaction. So there's a certain range of heights, but depending on nutrition and physical activity and things like that, that can influence that range of where you actually fall in the range. So all those are environmentally impacted. Even though you have a sort of a set range, the environment dictates where you actually end up in that range. And any of these characters that are influenced by both genes in the environment are called multifactorial, meaning there are many factors behind the actual end phenotype. And that's most of human characteristics, especially the ones we can see, are almost all multifactorial, where it's many genes involved. There's environmental influence. Turns out environmental influence probably changes the gene expression, and this equals different phenotypes. That's why it's really complicated to figure out human disease a lot of the times, because it is multifactorial. So you're looking at 50 different genes that are all influencing one phenotype, and on top of that, the environment influences how those genes are regulated, which gives you different phenotypes. And so to try to parse all that out takes a lot of time. And if we actually think about ourselves, we're not just our genotype, we're a record of our genotype plus our environmental history and how that's influenced how the genes are expressed. Which is one of the reasons even twins who have the same exact genotypes will have different phenotypes. And we'll talk more about that gene regulation that leads to those differences in the next unit.